Okay, thank you. Dr. Greek, TB, malaria, and HIV cause the death of millions of people every year. Without animal models, how do you expect to create vaccines to protect people against these diseases?
Well, that's a very interesting question, because just this week another HIV vaccine failed. It worked really well on monkeys. It's, I think, the third or fourth or fifth, and there's another 20 that are in clinical trials. The choice of infectious diseases in that question is also very interesting, because chimpanzees and monkeys do not suffer from HIV. Monkeys suffer from SIV, which is a completely different virus. Chimpanzees and monkeys, I believe, do suffer from tuberculosis, and malaria I'm not sure about. I don't remember. But this illustrates the problem. Here's 3 different viruses that cause at least 2 different reactions, completely opposite reactions, in humans versus humanity's closest evolutionary cousin, the chimpanzee. And in terms of how are we going to make vaccines in the future, pretty much the way we've made them in the past. If you want to know how the polio vaccine came about, there are some outstanding books on that, written circa 1970 by people who were actually involved in the polio vaccine. And somehow society has got this impression that the polio vaccine was dependent upon monkeys. And monkeys were used as incubators during that time. They absolutely, literally grew polio virus in the monkeys. But in terms of a model for polio, they were very misleading. And Al Sabin even said that the monkey model probably set the polio vaccine back about, I think he said 20 or 30 years. So how are we going to make vaccines? Pretty much the same way we've always made vaccines. It wasn't until polio was grown in culture that we finally got the vaccine, because then they could work with it and so on and so forth.
Eric?
I guess I didn't really hear an answer to that question. Pretty much how we've always made vaccines. I remember the first vaccinations, some of the first ones were that scabs from cowpox were rubbed into a cut in a human to immunize against smallpox. I don't expect that's what he means. There have been a lot of vaccines that have been developed in animals and then applied to humans. There's also another issue, and that's accuracy. And I think it's very important to be cautious about what you say. So it turns out that there was a vaccine study that was reported as failing this week for HIV/AIDS. The results in monkeys using the same vaccine, though, were not a success, as Ray said. There were 2 vaccines tried in monkeys. One, the monkeys were infected twice with a virus that contained some of the molecules from HIV. In another case, the monkeys were infected once with that virus, but the second time were administered naked DNA that is incorporated into the monkey cells and produces the antigen that serves as a vaccine. And as it turned out, the monkey trials showed failure with the 2 adenovirus infections, the 2 viral infections, and showed success with the adenovirus plus DNA. But the drug company, because it was so difficult to prepare the DNA, went ahead and performed the adeno, the 2 virus trial. So here is an example of where the monkey was predictive for humans. It failed in the monkey, it failed in the humans. And I think it's very important to get facts straight. And I think this is an important fact.
This could serve as a follow-up to that question, I think. Dr. Sandgren, do you believe we have lost drugs that would have cured diseases because they tested poorly in animals?
So whenever, and Ray and I agree on this, although certainly I think the extent of our agreement overlaps only very slightly. If you are relying on animals to give you information about humans, there are going to be times when you're not going to know whether that information is accurate or not. So I would say, absolutely, there have been cures that we have lost because we've tried them on animals and that they have not been efficacious or because they've hurt the animals. But then I would say, what's the alternative? Do you do large-scale toxicity testing on humans? There was a slide that Dr. Greek showed about toxicity in liver, and I can't remember whether it was mice in humans or a different species in humans. And he commented that the humans weren't really given large doses, just small doses, and yet the word toxicity was there. Well, that's a misuse of that term. Toxicity means damage, and I don't know of any institutional review board that reviews human studies that's going to allow testing to the levels where toxicity can be determined, where damage can be identified in humans. So, yes, we do lose some promising therapeutics. But the alternative is testing specifically on humans and having a lot of humans die. And even according to Dr. Greek, that would not be enough because humans are so different. So we're doing what I would say is the best we can.
So Dr. Sandgren and I do agree on the fact that we have lost cures for diseases, just like the National Cancer Institute and other people have said. There are numerous quotes throughout the literature that I won't bore you with, since Eric and I actually agree on this point. But I think your comments about the HIV vaccine are very telling. Now, I still maintain that I'm right on that, surprisingly enough. However, let's assume for the sake of argument that you're right. What does that say about the pharmaceutical companies' confidence in the monkey tests? If they're going to ignore the monkey tests and the data, and they're going to go ahead with this vaccine, even though it failed miserably in monkeys, boy, that sure looks like somebody at the pharmaceutical company doesn't believe in animal models either.
Okay, thank you. Dr. Greek, do you recognize the use of chimpanzees for HIV and AIDS as a failure?
Not only do I, but the current head of the National Institute of Allergy and Infectious Disease, or I may have that slightly wrong, he has recognized it as a failure. The chimpanzee researchers have recognized it as a failure. There is a very nice essay on the use of chimpanzees in HIV research in The Scientist, I don't know, probably 6-7 years ago. And the NIH recognizes it as a failure. To the best of my knowledge, and I'm pretty sure I'm right on this, there are no chimpanzee HIV studies that are currently being funded by NIH. Now, if it was a great model and if it brought us a lot of interesting data and so on, I think we'd be pursuing that.
And I'd say Ray illustrates very nicely the way science adapts to information. When scientists discover that something is not a good model, they abandon it, as they should.
Dr. Sandgren, isn't it true that many laboratories keep using animals because of companies that provide lower costs rather than moving towards non-human techniques? Not sure I quite understand that question. Thus, the breeding and selling animals is big business, so changes just don't happen.
I'm not sure I understand that either. Would you read that one more time?
Okay. Isn't it true that many laboratories keep using animals because the companies that provide, I guess, the animals are lower cost than, I guess, using other techniques that could be employed? Thus, the breeding and selling of animals is big business, so changes just don't happen.
So breeding and selling of animals is I don't know if you call it a big business compared to a lot of other businesses, but it is a business. I do know as a scientist that a critical aspect of performing science is matching the model that you use to the question that you're asking. And if you can answer a question without using an animal, that's the approach you're going to take. because it's far less costly. So there is a bigger expense to using animals in research than most other types of methodologies. That's why it's important for there to be strong justification for the use of animals in research.
Okay.
This will give me the opportunity to very briefly say why I think the animal model continues to be used, and you won't find science on any of the bullet points. The reason that animals continue to be used in science, yeah, there's a lot of money involved. That's true. But you really can't say that money is the only reason. I wouldn't even say it's necessarily the biggest reason. I think the biggest reason is tradition. Okay? The system is based on it. Okay? We've always done it that way. And I don't care if you're society or the FDA or the pharmaceutical company, there's a lot of resistance to change. Okay? And these are two very innocent reasons why the animal model persists despite its many failures. Now, there's some more nefarious reasons, okay? But the only point that I'm trying to make is that there are a lot of reasons why bad habits are not immediately dropped. Perhaps we could think of a war that we could use as an example. So there's a lot of reasons why it persists. It's not a very straightforward, simple, linear problem. Yes, money's involved, but there's a lot of other reasons.
Okay, Dr. Greek, question for you. If not animal research, you say there are several other options. Name three and give some statistics demonstrating their utility over animal testing.
I think that's a very good question. I don't have that data on the tip of my tongue. But if we would like to pursue that at another time, I think that's a very excellent question. So since I don't have data, and I'm not going to try and make it up, let me just say that the so-called alternatives, and I don't like that word, and I'll tell you why, but the so-called alternatives to using animals to study human disease is to use humans, okay? Not as guinea pigs, not in a human experiment like in Nazi Germany, but to study human beings in an ethical fashion. And I don't care, there's a lot of different ways to do that. Epidemiology is a way to do that. In vitro research is a way to do that. And we do that every time we take a surgical biopsy. We look at that tissue. If the tissue is interesting, we look at it a little further, and so on and so on. So human tissues from autopsy, from pathology, human genetics like these genome-wide association studies, plain old clinical research and clinical observation. Clinical observation has been sold very short in the importance that it has played in the development of medicine. And Eric asked me earlier, and I didn't have time to address it, why I said there's no alternatives to using animals. And a lot of the things that he said were taken very much out of context from my website. And I do encourage you to go there and look at what was on that slide versus what I actually say. I say there's no alternatives to using animals to studying human disease, just like there's no alternatives to using a used car to travel to the moon. You can't use a used car to travel to the moon, despite what the used car salesman will tell you, okay? And he will tell you that you can jump in that puppy and go to the moon tomorrow, but you can't. Alternative implies viability. And if we'd like to get the dictionary out, I think you'll find that I'm correct on that. So, no, there's not an alternative to something that doesn't work, okay? There's a lot of things that do work, and that's what we should fund. But I hope that answers your question.